discussing on this meiosis, right? We have discussed the different phases of meiosis. There is division 1 and division 2, you can say. Division 1, all the the phases, they are referring it as the phase 1, and then division 2, that like prophase 1 in division 1 and prophase 2 in division 2. So we have discussed this, but yeah, what all important things also we discussed, but it's very important to know, because many times you fail to understand this, how the mental is really, yeah. This what is important, division 1 and division 2. You just look at always in the mitosis, if you look at it, is having the prophase, metaphase, anaphase, telophase, and cytokinesis. That is the division of the cell. Otherwise, prophase, metaphase, anaphase, and telophase, all this division of the nucleus. After nucleus division, it is the cell division, that is cytokinesis. So these steps are there, but all significant difference from the mitosis and meiosis is this prophase 1. Otherwise, metaphase 1, it is same as metaphase 2. Anaphase 1, it is same as anaphase 2. And telophase 1 is same as telophase 2. That means it is same as the mitosis only. Whereas this prophase we were discussing, this is important and we have discussed. So there are the 5 phases of prophase 1. This only actually differentiates from the mitosis. So it is involved only in sexual parts, that is gamete formation. Otherwise, you don't find this meiosis in a simple asexual method of reproduction. This leptotin, that means the chromosomes are there in nucleus. It is everything is the very thin thread-like structures, but they start condensing. I told you that it starts condensing, then only you will be able to see under the microscope. So the membrane, these chromosomes start appearing. That is in lepton. Then zygote, it is the synaptonomal complex. That is homologous chromosomes, they start coming together. This is called the synapsis. Right? If they come and pair, is synapsis. We were defining terminologies like synapsis and asynapsis. By some means, some problem that asynapsis can happen, that means they are not pairing, they are not coming together. That is referred as asynapsis. Packeting phase, that is non-sister chromatids, they exchange that. Now homologous chromosomes, they come together in zygote. Now at the packeting stage, the non-sister chromatids, they exchange the genetic material. That is crossing over will take place. That means some fragment of the chromatid of first chromosome will be attached to the second chromosome, like that. They are homologous only. But yes, there is a genetic material difference is there. The DNA difference can be there. That's what your difference in the genes you are referring. So this cut and paste is happening. So that is non-sister chromatid exchange the genetic material that is crossing over. This is the origin of variability in the next generation, right? This crossing over is very important, which is going to give you variations. That means, they change because, non-sister chromatids, that means if that is, heterozygous condition is there, then there is a possibility of having the, in zygote formation that both dominant alleles coming together. So that's what it happens here. Or changes if it is, there can be, 2 different alleles can come together at the time of zygote formation. So this is coming together, zygote. Then the packetin is non-sister chromatids, they exchange the genetic material. The diplotin, synaptonal complexes vanish, and towards the conclusion, the diplotin synapses although the homologous pairs remain linked at the chiasm. So they start separating, but the thing is that the place where the crossing over the collectine paste has happened, only there they are linked, they start. The dikinesis means the chromosomes become fully condensed and the nuclear membrane and nucleus break down in preparation for the next phase of meiosis. That is the, the metaphase. Next phase is metaphase. So you just see the figures here, leptotin here and start condensing, here more condensed and pairing and this crossing over, and then they attached to only at the chiasma, and then they start, separating away. So this is the preparation of the entire in the nucleus membrane. It is getting dissolved and it is free. The entire cell will become prepared for the metaphase. Yeah, this is what the prophase. All the 5 phases are happening. Right? And then with the help of the spindle fibers, what you call the microtubules, the chromosomes, they are moving to opposite poles. And the sister, the 2 chromosomes, paired chromosomes, they move away. And anaphase, they are arranged at metaphase 1. They are arranged in equatorial phase. Only they are attached at the centromere or at the chiasmate where they are attached. Then they start moving away in anaphase 1. And telophase 1, yes, it is almost like the grouping into different, and the cytokinesis follows that the cell will divide. So one cell becomes 2 cells here at the meiosis 1, right? Then meiosis 2 is nothing but prophase 2, metaphase 2, anaphase 2, and telophase 2, then cytokinesis follows. So that means these two, whatever 2 cells are, that individually they divide, they duplicate. It's the same like mitosis only. It's a copy. Now one cell becomes 4 cells. That's called the tetrad, right? 4 cells you're getting. So in that process only, we got into these definitions, as I said. Synapsis, failure. Asynapsis means failure of pairing of homologous chromosomes during meiosis. Synapsis means conjunction at the packaging and zygotin of homologous chromosomes at that phase. Distinction, the separation of the chromosomes at anaphase. Let's start separating it out. Yes, well, how you can define this mitosis? Mitosis, a double, meiosis, it's a double mitosis. 2 times cell is dividing here, occurring in sexual reproduction, which results in production of gametes with haploid chromosome number, right? First phase is the reduction phase, because the number of, if there are 48 of 24 chromosomes, they are going to get into 2 cells. Let's see, let's go back. This meiosis phase 1, here reduction in chromosome number. For example, if you have 24 chromosomes you are starting with, the moving away, 12 chromosomes here. So each cell will have 12 chromosomes. But subsequent division is just duplication. That means this 12 chromosome, 2 cells will become. This one more cell of 12 chromosomes become another 2 cells. That means it's the same copy, 12 chromosomes only. So that means 12 chromosomes having 4 cells becomes. So it started with the 24. It is the reduction division. Here we are reducing the phase 1. Meiosis phase 1 is the reduction. The phase 2 is just a mitosis. So same thing is copied. That means it is divided. It is divided. The chromosomes, that copy is prepared, and they are formed into the gametes. Actually, 4. We are good. This is what the meaning of meiosis. That's very important for breeding. And of course, the Mendel's laws, whatever it is explained by using meiosis studies. Mitosis, the process by which a nucleus is divided into 2 daughter nuclei with equivalent chromosome complements, usually accompanied by division of the cell containing the nucleus. That is cytokinesis. The cell will divide. Then gamete, cell of meiotic origin. How do you define the gamete? It's a cell of meiotic origin, not any cell, right? Cell developed through meiosis. Cell of meiotic origin, specialized for fertilization. It's what is that? It is not going to survive on its own. It has to get fertilized, then only it can perpetuate, right? Otherwise, those gametes they cannot have individuals. Those are very, very rare chances, gametic cells directly getting into that plant, individual plants. They cannot, rarely. But yes, they are meant for fertilization. Then the basic number we said it, monoplide, you said it right. Where you said it, the monoplide is the haploid where it has the basic chromosome. The number of chromosomes in ancestral diploid of polyploid represented by X. There is no similarity of any of the chromosomes within that genome, that group, right? X. Bivalent, yeah. Is this pairing we said it? They pair in the first prophase, pairing a pair of homologous chromosomes united in the first meiotic region. Two are there, bivalent you call. And if there is no pair, single strand chromosome you are finding, you call it as a univalent. Or there can be trivalent and quadrivalents also. 3 chromosomes together, 4 chromosomes together. Those are all homologous chromosomes only. They are these univalent means univalent means it's a single. There is no pairing. Quadra means there is pairing, but they are attached at chiasma, right? They are crossing over wherever happens, the chiasmata, right? There only they are attached. There are various other terminologies we are getting into. Nulliplex, the condition in which the polyploid is now the condition is a polyploid you are referring, in which the polyploid is recessive in all chromosomes in respect of a particular gene. Right? All the the chromosomes, right? Your polyploid is there, maybe triploid, tetraploid, hexaploid, whatever. All these chromosomes, you are referring to one particular gene. That particular gene in all the chromosomes, it is having a recessive allele, right? The condition in which polyploid is recessive in all chromosomes in respect of a particular gene. Simplex denotes recessive at all chromosomes except one, right? Except one. And the duplex means there are two recessive. Simplex means, it denotes the recessive, duplex denotes the recessive at all the chromosomes except one. Duplex means all except 2. Triplex means 3, like that. Quadruplex. So how many, it is having? It's all. Nulliplex means nothing is dominant. Okay. Simplex means one is dominant. Duplex means 2 chromosomes where the dominant allele is there. 3 dominant alleles. And then triplex, okay, quadruplex means 4, like that it can be. So rarely you can encounter those things. So duplex, nulliplex 0, simplex 1, duplex 2, triplex 3, quadruplex, or SC polyploid, except particular loops, that particular gene. That is nulliplex, simplex, duplex, triplex, quadruplex. You are referring to one particular gene, and all genes are having recessive. The nulliplex 0, simplex 1 dominant, duplex 2 dominant, triplex 3, like that. Now the term like nullizome. What do you mean by nullizome? So if you study euplites and aneuploids, you'll clearly understand. I hope you have studied. But straight away, if you are asked to define what do you mean by this nullizome, it's a 2N plant that lacks both members of one specific pair of chromosomes, hence with 2N minus 2 chromosomes. That means one particular pair of chromosomes is completely lost. For example, in tomato, we have got 2N is equal to 24. We know that 12 pairs are there, right? So the 2 chromosomes, that means it's having only 11 chromosomes, 11 pairs of chromosomes. So the 12th pair is completely lost. That is 2N minus 2. So that is called nullizome. Completely vanish those genes whatever those chromosomes are carrying, it is not there, the individual, right? But now you are referring to the individuals which is having this condition, nullisome. Then monosome. Now, so the nulli means complete pair of chromosome is lost. Monosome. In organisms, these are all aneuploids, right? Aneuploids were defined. An organism lacking one chromosome of the diploid component, hence having 2n minus 1 chromosome. So one particular chromosome is not there. So that means one chromosome is there in one only. That means it has to be diploid, right? Pair should be there. Instead of pair, single chromosome is there. So for example, for your tomato, again, you take 11, 2 pairs are there, 12th chromosome, only one is there. One more should be there to become a complete diploid, but it's lacking that one chromosome. That is, the individuals having that kind is called as organism we are referring to, remember. Right, this organism and how it survives and all, it's going to happen, not always. Very few cases where, it has been studied, they are, the aneuploids, nullisome. Similarly, trisome, an organism diploid except for one kind of chromosome, which is present in triplicated, now additional. Hence, for example, tomato, you have got 12 pairs of chromosomes. The 12th pair is having one more additional chromosome. So 2n plus 2 chromosome. That is trisome. Additional chromosome is there. Tetrazome. An organism diploid except for one kind of chromosome, which is present in quadruplate. 2 additional chromosome. Right? 2 additional, 2n plus 2. So 12 pairs of chromosomes in tomato. The 12th pair is having another 2 chromosomes. That 12th chromosome is having 2 additional chromosomes. That is 2n plus 2. Tetrazome. So nullisome, complete pair lost. Monosome, one extra. Trisome, sorry, one less. Monosome, trisome, one extra. Tetrazome, 2 extra. So these are the conditions you can get. Then the translocation, change in the position of a segment of chromosome. This is all again chromosomal aberration results only. That's malformation at the time of meiosis. Change in position of the segment of the chromosome to another location in the same or a different chromosomes. Right? Change in the position of a segment of a chromosome to another location. The fragment of the chromosome is cut and present some other place, right, on the chromosome. The same or a different chromosome. Maybe it has got some other. So that is translocation. Not lost, but it is changed to some other chromosome, that fragment of the chromosome. Inversion. It is the rearrangement of the chromosome segment to that its genes are reverse linear order. Order is reversed. That your strand is there, it is just cut and reversed and then paired, the pasted there. So inversion, rearrangement. If you give the order of the genes there, it gets reversed. Position of the gene is changing. That's all. These things can happen at the time because of the chromosomal operations. Maybe various reasons. What do you mean with the chromosome? The structural units of the nucleus which carry the genes in linear order. Whenever it is asked, you should be able to define any term correctly. Maybe you have understood, but defining at the time of interview or any person is asking you, so that person will understand, oh, you know that. Otherwise, although you know, you do not express it properly. So they assume that you are not having the knowledge about this. That's why this, the sentences of definitions, very important, meaningful sentences. Structural units of the nucleus which carry the genes in a linear order. The chromosomes undergo typical cycle in which their morphology change drastically in various phases of the life cycle of the organism. You have seen, that's very thin, nowhere you can find, they start condensing, you can observe under microscope, changes happen. Chromosomes are getting changed. There may be the chiasmata, separation of the chromosomes and chromatids, all that it can undergo.. The chromosome means always there are two threads which are attached to the centromere. one of the two thread structures, one of the two thread-like structures formed by the longitudinal division of the chromosome during meiotic prophase and known as daughter chromosome during anaphase. Okay, just a minute. That is chromatid. This also, the one of the two thread-like structures formed by longitudinal division of chromosome during meiotic prophase and known as the daughter chromosome during anaphase. Kinetochore or centromere, the spindle attachment, the place where, right, localized region in each chromosome to which the spindle fiber appears to be attached and which seem to determine the movement of the chromosomes during the mitosis and meiosis. This is the centromere, center place, right? Where only spindle fibers are attached, you have seen. So two chromatids are attached at the center. That center place is called as the, the centromere or kinetochore also. Chiasma, chiasmata, we said it, an exchange of partner between paired chromatids in the first division of meiosis. Non-sister chromatids, when they pair, they exchange, they cut and rejoin. An exchange of partners between the paired chromatids in the first division of meiosis. Then, yes, it is the crossing over, the exchange of corresponding segments between the chromatids of homologous chromosome during meiotic prophase. In genetic consequence is the recombination of linked gene. So normally the crossing over, if it happens, definitely you will get the recombinations. For example, if one gene in the dominant condition, another gene is in recessive condition, if in between the crossing over happens, then that dominant and recessive, what you say linked, or that gets separated. That means you can find both dominant or both recessive also. That means breaking of that. So in the genetic consequences is the recombination of linked genes. The linkage, it's because of the proximity of the genes. They are near, the crossing over can happen any place. If they are very near, the chances of having crossing over between those two near genes, it is very rare. That's why we start calling it as a linkage. The linkage is association of characters in inheritance due to the location of gene proximity on the same code. I told you, if they are very near, if the crossing over is not happening, then always one dominant, another recessive, or both dominant genes, they always inherit. You don't find one dominant and another recessive, or both dominant, or both recessive condition. That's because of the proximity of those genes. So now if you are referring to the characters, and those characters are controlled by genes, and those genes are located very near, and crossing over is not happening, and you start calling it as a linkage. The association of characters in inheritance due to location of genes in proximity on the same chromosome. It's always, naturally, it has to be on the same chromosome. And again, near the place, location is also very important. Very near, then only linkage you can find. So the association, that means that those characters tend to go together. For example, some pigment is associated with the hairiness on the leaves. That means the genes controlling that pigment and also that hairiness on the leaves, they are located very near, and always the pigment is there means you will get a hairiness. So that's the meaning of linkage. Linkage map of a position of the genes in chromosome determined by recombination relationship. The frequency with combinations are happening. Based on that, you can determine the distance between the chromosomes. The genes on the chromosome. That is, put in the form of map, linkage map. Linkage value, right? Recombination fraction expressing the proportion of the crossing over versus the parental types in a progeny. The recombination fraction can vary from 0 to 1.5. How frequently that crossing over is happening between those genes? You're terming it in the form of a value that is known as a linkage value. So recombination fraction expressing the proportion of the crossovers versus parental types in a progeny. The recombination fraction can vary from 0 to 1.5. So that is the value how frequently are going to get. Yeah, linkage value. Then the coupling phase. Linkage and coupling phase, they say. The linkage means it's two genes are linked. So what is that coupling phases you mean? Linked recessive alleles occurs in one homologous chromosome and their dominant alternative occurs in other chromosome. That means both the genes or recessive genes are linked. That means dominant genes are also linked. You don't find one dominant and another gene recessive. So that is coupling phase, right? Both genes, recessive or dominant, are occurring together, right? The linked recessive allele occur on one homologous chromosome and the dominant alternatives occur in another chromosome, right? So this is coupling phase. Linkage and repulsion phase, in which one dominant and one recessive occur in each member of the pair of the homologous chromosome. That means first gene dominant allele is there, second gene recessive allele is they are together. Vice versa. Second gene, the first gene recessive, second gene dominant. First gene dominant, second gene. So like that, it is one is dominant, other is recessive. That combination only it is forwarded from generation to generation because they are. So that is called as a repulsion phase. One gene is dominant condition, another gene is in recessive condition. They are there together, right? One dominant and one recessive occur in each member of the pair of chromosomes near. So proximity, then only the linkage. First the linkage and then two types. Coupling phase, both recessive are dominant. Repulsion, one recessive and one dominant. Always, linkage means you are taking two genes together and explaining it. Right? Both genes recessive are dominant, coupling phase. One recessive and one dominant, you are referring it as a repulsion phase. Pleiotropy, when two characters are controlled by same gene, and it is often due to tight linkage. When two characters are controlled by same gene, and it is often due to tight linkage. Pleiotropy means one gene is controlling two characters. So sometimes you may be under illusion that same gene is controlling. If the two genes, those two characters are controlled by two genes, but they are tight, they are placed very closer. No crossing over is happening between those two genes. Then also it leads to pleiotropy. That means one gene controlling two characters. You're saying when two characters, when two characters are controlled by same gene, and you will get confused due to tight linkage of the two genes. Parthenogenesis, the development of an organism from egg cell without fertilization. Right, egg cell is a female gamete, right? But it is not undergoing the fertilization, but the individual you will get, the plant you will get from that. So that organism you are going to get. Development of an organism from egg cell without fertilization. Okay, so that is the art. That art and yes. What do you mean by the breeding? The art and science of changing plants or animals genetically. That's it. That's it. That's how you can define. The art and science of changing plants or animals genetically is breeding. Genetically or altering, that is breeding. Line breeding, a system of breeding in which number of genotypes which have been progeny tested in respect to some characters or a group of characters are composited to form the variety. A system of breeding in which number of genotypes which have been progeny tested in respect to some characters or a group of characters are composite to form a variety. Is that it? The line breeding means why that's a progeny testing you are doing. One from one plant you are collecting seeds and raising a separate row. All the plants in that row, they are really doing it. Like that, you have tested several such genotypes. And with the help of progeny test, you are validating the superiority of that plant. And such plants, you can push it as a variety. You can say that breeding procedure is the line breeding, a system of breeding in which a number of genotypes which have been progeny tested in respect to some characters or a group of characters are composited to form the variety. Okay, that's line breeding. Breeder seed, nucleus seed produced by the agency sponsoring a variety and used to produce the foundation seed. Breeder who has developed, or the institute which has developed, they are producing the seeds of that variety first. It's the first stage, you can say. Then certified, the seed used for commercial crop production produced from the foundation, registered or certified seed under the regulation of a legally considered agency. That's the foundation seed. After the breeder seed or nucleus seed, it gets into the foundation seed, the seed chain. The seed stock produced from the breeder seed only. Right? Breeder seed. By or under the direct control of an agricultural experiment station where it has developed, or okay, that or some agency you can have. Breeder himself can control, or the institute where it is developed also can control. So the seeds used for producing the foundation seeds are breeder seed. The foundation seed is the source of certified seed, right? From the foundation, you can produce the certified seeds. So this is the foundation seeds are used for producing the certified seeds, either directly or through the registered seed. Truthfully labeled seeds, most of the seed companies, they do this. Seeds produced and labeled by individual, organization, and solely responsible for consequences of food quality. If anything happens, they are themselves are responsible. They own that seed if any problems are there. Otherwise, the seed producing agents can take the responsibility. If they certify, that means yes, under our vigilance, it has been produced, and legally that organization can help you. Any problem arises. So, roguing, removal of undesirable individuals to purify the stock, right? Undesirable individuals. Rogue, the plant, the plant showing variation in expression of distinguishing characters or abnormal performance of the plant in comparison to the normal expression of the cultivar, that is a plant of other cultivar, other species, or disease plant of the same cultivar, you can refer it as a rogue. Off type, a plant in the seed crop which deviates from the norm for the cultivar, but does not obviously belong to another cultivar. Plant is similar for distinguished characters, but deviates in agronomic traits like days to flowering, plant height, disease reaction, and days to maturity. That means maybe that variety while developing some amount of impurities there, or some quantitative characters, they are being influenced by the environment, like it is said, it is agronomic characters, it deviates, but it doesn't obviously belong to other cultivar. Off type. Rogue means it is more dangerous. Off type means okay, a little bit near to that. That's the meaning, right? Near to your variety. Yeah, combining ability. We often use the term. General combining ability and the specific combining ability are the two types. General combining ability is average performance of the strain in a series of process. Combining ability is not its own ability. Remember, it's like a progeny testing only. The progeny is how they are performing. Based on that, you are saying that genotype combining ability, how it is, good combine or not. So again, the two types, general combining ability and specific combining. General, average performance of the strain in a series of process. One genotype, about which you are trying to investigate on the general combining ability, it will be crossed to several genotypes on the average of the progenies of those several genotypes used as parents, second parent. Average of those efforts, performance is known as general combining ability. Whereas specific combining ability, it is, remember, most of the time many of the people, I mean, students, they do not know this, but still, something they said, they just try to derive the meaning from the literal meaning of the word, like general, specific. See, you have to be very specific here. Specific. It is deviation from the performance of the predicted on the basis of general combining ability. So specific combining ability means it is dependent on the two parents, whereas general combining ability, it is referring to one parent, whereas specific combining ability is referring to two parents, parent 1 and parent 2, right? Now, the parent 1 and parent 2, if you cross, get a F1 hybrid, but parent 1 and parent 2 can be predicted based on their general combining ability. On the deviation from the general combining ability of the parent 1 and 2, right? Deviation is known as a specific combining. Deviation from the performance of predicted on the basis of general combining ability. That means the performance of the F1 minus the general combining ability of the parent 1 minus the general combining ability of the parent 2. The remainder is the deviation. So that is your specific combining ability. Remember, specific combining ability, you are referring to two parents. General combining ability, you are referring to one parent. How you are deriving that general combining ability? That's the one parent is crossed with so many, and the hybrids you are getting so many. The average performance that hybrid is nothing but the general combining ability of the parent. Similarly, like any genotype, you do it. But specific combining, now it is you are referring to two genotypes. Two parents are involved in crossing. So the two parents are involved and you get a hybrid, and that hybrid performance, if it is deviating from the performance predicted based on the general combining ability of parent 1 and parent 2. Okay, that is specific combining. Dialyl cross. There are different types of dialyl crosses, like complete the crossing in all possible combinations of series of genotypes, right? Your permutation and combination. Dihybrid, heterozygous with respect to two genes, right? You are referring to that. Two genes, dihybrid. Single cross, a cross between the two genotypes, usually two inbred lines in a plant breeding. Double cross hybrid, cross between the two F1 hybrids, two single crosses. That is double cross. Dihybrid is different from double cross hybrid. I'm just placing these terminologies together so you should not get confused. Dihybrid, you are referring to the two genes. How many genes? Two genes. It is heterozygous, dihybrid. Double cross hybrid, it is the cross between the two single crosses. Single cross, it is a cross between the two genotypes. Triple cross, cross between F1, one hybrid, and the third one. That is a triple cross. Okay, hybrid 1. I mean, the hybrid 1 is derived from parent 1 and parent 2. You get a hybrid. That is crossed with the third parent. That is called the triple cross hybrid. So three-way cross hybrid, cross between the two triple cross hybrids. You have crossed the parent 1 and parent 2, you get a hybrid. That hybrid is crossed with parent 3, that is triple cross. Similarly, you are crossing the parent 4 and 5, you get a hybrid, that is crossed with the parent 6, you get a hybrid. So that is, it's a second triple cross hybrid. So triple cross hybrid 1 is crossed with triple cross hybrid 2 is called a three-way cross hybrid. Cross between the two triple cross hybrids. Test cross. A cross of a single or a double or a multiple heterozygote to the corresponding recessive parent to test the homozygosity or the linkage. Test cross hybrid. The cross of a single or a double or a multiple heterozygote. Now, test cross always you do it for, homozygosity. So hybrid is crossed with the one of the parent, right? A hybrid is having the heterozygosity for one gene or two gene, whatever. What? One. Heterozygous for 1, 2, double, or multiple. It's crossed with another parent to test the homozygosity. So, for example, F1 is crossed with the recessive test cross. You are resulting with 1:1 ratio. So predicting, right? Test cross ratio predicting. Top cross terms are used, right? It looks same, but they are different. Top cross, a cross between the selection, line, a clone, etc. Anything, right? With a common pollen parent, which may be a variety in bed line, single cross, etc. The common pollen parent is called the top cross or the tester parent. Okay, cross between a selection or a line, clone, etc., and a common pollen parent, right? So, which may be this common pollen parent can be a variety, it can be inbred, it can be single cross. So, the common pollen parent is called as the top cross or the tester parents. So, cross between the tester parent and which you want to select the line, right? So, cross between the selection with a variety, right? That is a top cross. Polycross, open pollination of a group of genotypes. I just explained in the previous class. Open pollination of a group of genotypes generally selected in isolation from other compatible genotypes in such a way as to promote the random mating intersect. So one plant is placed in the center and you are collecting the OP seeds surrounded by so many different genotypes that OP seeds of that one plant is containing the polycross. That means single plant is producing so many hybrids because of the pollen parent variation, whereas female parent is same. So that is referred as polycross. You are not going to cross many crosses or like something like that. Okay, open pollination of a group of genotypes in isolation from other compatible genotypes in such a way as to promote the random mating intersect. It's always reciprocal cross in which the source of male and female gametes are reversed, right? Okay, the gametes, the pollen parent and the ovule, you are interchanging parents. Panmixia, random mating in a population without restriction, usually extended to include random mating under the restriction of sex or incompatibility, right? Without any restriction, random mating. It's open pollination. Gene frequency, the proportion in which the alternative alleles of a gene occurs in a population. You said it, there are two alleles of the particular gene, capital A and small a, right? Now, if you analyze the entire population, there are 100 plants. Each plant is having two alleles for that particular A gene, right? Now, how many entire, 100 means 200 alleles will be there for that. So out of these 200, how many are dominant and how many are recessive? That is the frequency. Okay. So the proportion in which the alternative allele of a gene occurs in the population, right? So out of 200, now 100 plants, diploid plants are there, means 200 alleles are there. Out of these 200, how many dominant and how many recessive? And if you get a ratio of that dominant to recessive or recessive to dominant, that is referred as a gene frequency. So gene frequency ranges from 0 to 1. Ratio of dominant allele, gene frequency, right? Ratio of dominant alleles, that's referred as P, to recessive, that is Q in a population. P by Q, where P plus Q is equal to 1. Okay, so P plus Q is equal to, so ratio of dominant allele is P and ratio of recessive allele is Q, right? Right? So if you add the ratio of dominant allele plus the ratio of recessive allele, it is equal to 1. Okay, I said it, 200 alleles are there. If there are 60 dominant alleles and 140 recessive alleles, what do you mean by 60, right? So now the gene frequency, the total genes are 200, 60 divided by 200 and 140 divided by 200. So you get a ratio. If these two ratios, if you add, it will be 1. Okay. So genotype frequency, that is gene frequency you're referring. Genotype frequency, a ratio of a given genotype to total population in a random mating population. As I told you, the gene, it is dominant and recessive, A gene, like capital A and small a, and gene frequency. Now, instead of that capital A and small a, now I have a genotype. That means if it is controlling the plant height, that A gene. Both are dominant allele. We take that tall is dominant to recessive. I mean, dominant to dwarfness. So tall plants, how many are there out of 200 plants? How many tall plants are there? You make a count. And how many are dwarf out there? Now the ratio you will get, right? That ratio is given as ratio of a given genotype to total population in a random mating population. So that means out of maybe there are 50, 60 tall plants are there out of 100. So 60 divided by 100. What is the genotype frequency? Okay. If you take the dwarf plants, 40 divided by 100 dwarf plants. Yeah, this is what just we can take this dominant alleles P square, it's the heterozygous PQ, and homozygous recessive Q square. P plus Q square is equal to P square plus 2PQ plus Q square. So your mathematical derivation you can get into this frequency, genotype frequency also. So gene frequency means dominant and recessive how many are there. Genotype frequency means tall and it depends on how the intraallelic interaction is there. Drift, change in gene and genotype frequency in a small population due to random process. Sudden change in the gene frequency, right? Genetic drift, change in the gene frequency due to sampling error in a small population leading to allelic erosion. Some alleles will disappear if they are rare. Genetic drift. Genetic equilibrium, the condition in which the successive generations of the population contain the same genotypes, the same proportion with respect to a particular gene or a combination of genes. So it remains static, equilibrium it has reached. Very good. Then mating system. Any number of schemes by which individuals are assorted in pairs leading to sexual or reproduction. Mating systems referring a random assortment pair is by chance. Anything can happen. So any number of schemes by which individuals are assorted in pairs leading to sexual reproduction. Then genetic assortative mating, mating together of individuals more closely related than individuals mating at random. So assortative, similar types of mating. Genetic disassortative mating, mating together of individuals less closely related than individuals mating at random. That means it's not a random mating means anything can happen. Whereas assortative mating means now closely related are mated. Whereas disassortative means closely not related one are deliberately mated. So that is genetic disassortative mating. That is the genetic version. Now the phenotypic assortative mating, mating of individuals more alike in appearance than individuals mating at random. Phenotypic disassortative mating, mating of individuals less alike in appearance than individuals mating at random. I just said it, whites marrying white, phenotypic assortative mating. Whites marrying dark, phenotypic disassortative mating. The random mating, anything can happen. There is nothing, systematic. It's a sort by chance. Then inbreeding, the mating of individuals more closely related than individuals mating at random. Inbreeding, now related one are being, crossed. Then the individuals mating at random. Then inbreeding coefficient, a quantitative measure of the intensity of inbreeding. Okay, how that inbreeding. This is the other side of this is the outbreeding. Inbreeding depression. So by doing this, you got inbreeding, but by this you are observing in the progeny. It's the loss of vigor and physiological efficiency of an organism on inbreeding, leading to reduction in size, fecundity, etc. So that's a depression. Some loss is there in the progenies because of this inbreeding. So inbreeding how you work out the inbreeding depression, inbreeding coefficient. F2 minus F1 divided by F1 into 100. F2 minus 1. Now, next generation I got, and F1, if you're comparing, and the difference divided by F1 value, you into 100, what will be, you get the percentage. Okay, that's how we use very oftenly inbreeding depression, inbreeding coefficient. Sorry. Inbred line. A line produced by continued inbreeding. In a plant breeding, a nearly homozygous line usually originated by inbred line. A line produced by continued inbreeding. In plant breeding, a nearly homozygous line usually originated by continued self-fertilization, accompanied by selection. It is normally done in, the cross-pollinated crops. Inbred lines, you call in cross-pollinated crops only. Continuously are selfing, and the progeny is near homozygous. That means not complete homozygous. Inbred lines are not like pure lines, remember. Pure lines, a strain homologous at all loci, complete homozygosity. Homozygous at all loci, ordinarily obtained by successive self-fertilization in plant breeding. This is found in self-pollinated crops. Okay, this is found in self-pollinated. So you use the term pure line in self-pollinated crops, whereas inbred lines in cross-pollinated crops. For heterozygous breeding, in cross-pollinated crop, we should not use the term pure line. It's not correct because you don't get the pure lines. It's homozygous for the old loci. But yes, majority, nearly homozygous inbred line. Then isogenic line. What do you mean by isogenic line? Two or more lines differing from each other genetically at one locus only, right? Distinguished from clones, homozygous lines, identical twins, etc., which are identical at all loci. So, if you compare the two, they are alike in all the genes except one. Two or more lines differing from each other genetically at one locus only. Sorry, it is only one locus, right? So if the two individuals you are comparing, both the individuals are identical in all the genes except one. That is called isogenic lines. You compare more lines than two or more lines. So distinguish, they are different from the clones, homozygous lines, identical twins, etc., which are identical at all. Isogenic lines are very important for genetic studies, how the difference is there. Synthetic variety, a variety produced by crossing inter se a number of genotypes selected for good combining ability in all possible hybrid combinations with subsequent maintenance of variety by open pollination. Now, what you do is synthetic variety, you select the lines based on their combining ability and you grow them together and collect the seeds, OP seeds. That is your synthetic variety. Okay. What is the thing in the composite variety? Composite variety are not assessing it for the combining ability. Only in synthetics you are assessing combining ability. Progeny, however, right? Composite varieties, some good performing varieties you mix together. Whereas here, their progenies, their all possible combination progenies, their combining ability is tested, found superior. Only those are mixed to produce the seeds of that variety, synthetic variety. Composite is different, where the combining ability is not. Transgressive segregation, you please make the corrections, spelling corrections. Transgressive, not transgressive segregation. Appearance in segregating generations of individual falling outside the parental range in respect to some of the characters. Parent 1, parent 2 across for a particular character we are referring. If that offspring or the progeny is falling outside the range of the parent, That is called transgressive segregation. Something which totally unexpected or falling outside the range of the parents. SIB mating, the mating of SIBs. So what are SIBs, you know? Those parents are common, right? Again, SIBs means it's not full. Full SIB means both the parents are common. Half SIB means one parent is common. Full SIBs. Progeny of the same parent derived from different gametes. Same parents, how they are different? Because the gametes are different types of gametes are formed from the one parent. That's why they are different. But they are coming from the one parent. Full SIB, both the parents are common. Half SIB, one parent is common, another parent. Half SIB progeny with one parent is common. Protandry, maturation of anthes before pistils, right? So male is maturing before the female part. Protogyny, maturation of the pistil before anthes. Prepotency, the capacity of the parent to impress characteristics on its offspring so they are more alike than usual. Right? Prepotency, when you can predict the capacity of the parent to impress characteristics on its offspring so they are more alike than usual. Right? So then progeny test. A test of a value of a genotype based on the performance of its offspring, produced on some definite system of mating. You can, and based on selfing, it can be, or based on crossing also. Virulence, capacity of the pathogen to incite a disease. Okay. So virulence, virulence, capacity is, virulence means, yeah, it can cause the disease. Virulent. Virulent means it will not be able to cross the. It is the race of the pathogen, right? Then genia, effect of pollen on embryo and endosperm. Bitterness in cucumber, right? Some fruits are bitter and some other fruits are not bitter. How genetically you can explain? Because the pollen, the bitter pollen has come and fallen and the fruit get fertilized. I mean, the fruit is derived from the fertilization of the other pollen parent, which is bitter, is resulting into bitter fruit. So, genia, effect of pollen on the embryo and endosperm. Metagenia, influence of pollen on maternal tissue of the fruit, right? The maternal tissue of the fruit. So you have this, why it's very, see, some seeds are red, some are orange, some are white. Why? Because of the pollen parent is different. On the same, the cob of this maize, they are getting. So within a fruit, it happens. Whereas in case of cucumber, what is happening? The entire fruit is getting bitter. So the xenia and metagenia, the maternal tissue is getting, right? So this is, the on the seed itself, it's a genia effect, you can say in case of maize. Okay. Okay. So with that, we are completing on the definitions, but still I suggest you to go through the glossary and clear yourself on the different terminologies used in genetics and plant breeding to get yourself prepared for this course, Principles of Vegetable Breeding.